Welcome back to the cell culture technology. So, the first class is our second class of the first week. The first class I told you, when we take a part of the tissue and dissociate them and grow the cells outside, or even the tissue as a matter of fact, one of the biggest challenge is how close are we to the in vivo setup or the conditions what a cell experiences inside the body. Just to appreciate that, so let us start it today. So, we are into week 1, class 2. Okay. So, what we are asking is simple. So, say for example, so this is say this is an intact in vivo system, and say for example, I took a part of the cardiac tissue outside. Okay. This is the heart, and took part of cardiomyocytes. Cardio means heart, myocytes means myo means muscle, cardiac muscle, and cytes means cell. Those of you who do not have the wide range of biology background, this is the breakup of that word, cardiomyocyte. Cardio means taken from the heart. Okay. So, I take part of this, and these cells kind of look like this. And there are connectors called gap junctions. I am not getting into the detail of it at this point, like this. Now, I take these tissue, cardiac tissue, dissociate them into cardiac myocytes and allow them to grow in a dish. I have dissociated them, as I mentioned you, and allow them to grow. Once I allow them to grow outside the system, what do I expect from them? The first thing what do I do expect from them is to regain this tissue level cytoarchitecture. When I talk about the tissue level cytoarchitecture, they should develop these specialized structure called gap junctions. Gap junctions are small tube-like structure. Those who are from biology, they are aware of. Those who are from non-biology background, it's simple. Say for example, there are, just imagine it like there are two rooms. There is a room here. There is an adjacent room. Say for example, let me draw it just to make it simple. This is one room and this is another room. Okay. Now, if I put some connector like this, so these two rooms can crosstalk with each other, right, like this. These connectors are called gap junction in technical term, and these are nothing but these are proteins. These are membrane proteins which are forming these kind of junctions. Okay. So, cardiac cells have this classic feature. Cardiac tissue has this classic feature. They have series of gap junctions, and there is reason for it. We will come later into that. Why those gap junctions are important, because it is a structure where the electrical stimulation has to spread out in multiple directions like this, and it has its own function. And of course, there is one more thing. This gap junction may be sometimes gated or non-gated. They may only open in one direction, or they may not. They may have both directionality. Okay. So, the first thing what I anticipate for these cardiac cells to join with each other, coming close and form those gap junctions. So, essentially what I will be expecting next to happen is this. Here I will have these cells coming close like this, like this, like this, okay, and forming those, reforming those gap junctions. Are they doing that? Now, next thing, when you talk about cardiac, you touch your heart, it is beating. Okay? This beating means these cells, what you are seeing here in your body, these cells are essentially, they are performing two functions. They are forming a electrical function and a mechanical function. When I talk about mechanical function, that means what I was telling you is the beating. And this electrical function and the mechanical functions are coupled with each other. It's not these are independent things that, you know, if I am doing this, I will not do this. They are all interlinked with each other. If there is an electrical impulse, it will lead to the mechanical vibration or beating, whatever you call that. So, for these cells, if they have to grow outside, and they should be in a position which should be able to mimic this electromechanical event in a dish. So, these cells I expect now, they should be able to do that electromechanical activity similar to that of in your body. It should be similar. If it is not similar, then we know there is a deviation and we will have to quantify those deviations. Okay, it is not that that is a negative thing, it is a very positive thing. Then we know that we have a room to improve, and I will tell you why these are important, because these are really, really important for those people who dream of artificial organs. I mean, futuristic world where they say we can replace the heart. Yeah, sure, but if we do not know the basics, we won't be able to achieve that goal. Or people say that we can replace a kidney, we can replace something. I mean, there is the whole buzzword all over this tissue culture world, or regenerative medicine. But the fact is, this all lies in the basics. If the basics are not clear, these are fancy dreams. As a matter of fact, one is living in a fool's paradise without knowing the basics. Okay, so we have to really know what all you can achieve, and I'll tell you that why at times people just get allured without realizing the ground realities. There are certain ground realities. Okay, so it will generate an electromechanical activity. Now, this electromechanical activity, your first evidence, you should be able to see on a dish under a microscope that these cells are beating. Okay, they are not beating, it means, yeah, you grow something, but they are not functional. Now, if they are beating, it means they are generating certain force, right? It is a mechanical event, there is a force generation. Now, whenever we talk about force, we are talking about some Newton value, right? Something. So, what is? It could be nanonewton, it could be piconewton, it could be whatever. So, there has to be some unit. At what rate, at what unit this is mechanical activity is, because that will again tell you that how close you are to the real life. My heart is beating. So, part of my tissue which is going outside should exactly behave the same way. Now, if we have to correlate this, then we have to understand these beatings are governed by the two major proteins of muscle, which are, now let me put them in, those are actin and myosin, which I am putting in two different color, orange and green. Okay. So those are actin and myosin filament. And as a matter of fact, depending on the quantity of force generation, the myosin filament subtype changes. In other words, what I mean to say is the myosin present in these muscles of my body, which are skeletal muscle, are entirely different force dynamics than the force dynamics of the cardiac cells or cardiac myosin which are present there. Not only that, within this muscle there are certain structures called muscle spindle. They behave entirely differently. Their myosin properties are entirely different. Similarly, there are myosins in your gut, the whole smooth muscles which is lining the whole or which is forming the whole alimentary canal, where you are eating the food and the food is moving through a peristaltic movement across the digestive system. Those myosins are different. But it is that myosin, critically, actin does play a role, but it is this myosin subtypes which dictates, say, type, whatever, fast type, slow type, whatever. So, I am just putting A, B, C, just for imaginary sake, I am putting it like that. So, these cells which are growing in the dish now should express those myosins which are really present here. Then only there is a possibility that they will be generating the similar force as generated inside the system. So, you are realizing that when you are trying to build something outside the system, so this is something, suppose if this is on earth, you are trying to grow something, I mean if you are growing something, this is a totally different environment for it, it is in space, totally different, it has no clue. Why it has no clue? Think of it for a minute. Now this heart which is here, it is surrounded by, it not only heart has only cardiomyocytes, the heart consists of endothelial cells, then there are cells which are forming the valves, there are blood vessels, there are sympathetic and parasympathetic nerves, or rather autonomic nerves. There are stem cells, cardiac stem cells which are present there. So, these cardiac myocytes, what you have isolated, are coming from a milieu of, if not, I mean it comes from way more than several surrounding cells. So, it means you can imagine it like this, as a cartoon way you can imagine it like that. Suppose there are houses, okay, I am just drawing few houses for you and that will help you to appreciate it. Okay, you are seeing different colors houses what I am drawing through. Okay. Now, it is a kind of a colony where every house have different color, and yet some houses which are of similar color. So, you see red, you see light green, you see dark green, you see Persian blue, likewise. Now, what I am asking is that I only wanted to study these houses in isolation, and that too not in this system, outside this system. Now, the thing is that one can study, absolutely no problem, but one has to keep in mind that there is always an interaction between this, between this, between this, between this. There is an interaction between these ones, there is an interaction between these ones, there are interaction between these ones, there are interaction between these ones, these ones. So, when you are trying to study these red houses in isolation, you will be missing, there will be signals which you are missing, and you have to accept this error. It is not an error, rather it is an acute system you are growing. So, you may not be able to achieve the exact perfection, it is not possible. I mean, it is accepted because I do not know these many interactions which is happening, but this gives me an opportunity when I am growing this in isolation. So say for example, just try to take your imagination slightly beyond it. Say for example, I give an hypothesis, I say these pink color houses, you see these pink color houses? These pink color houses are communicating with these red color houses by a compound called, say, by some means, say X. Okay? So, this is my hypothesis. I said okay, fine, and I said that when X is sent to these red houses, the red develops something, say, develop another roof like this. So, fine, if this is my hypothesis, how I can prove this hypothesis, whether this is really true or not. So, what I do, I have to first of all figure out what is this X compound, okay? And I grow these in absolute isolation, and then in this I introduce that X, and I wanted to see does the X develop that additional roof or not. If it does so, that means whatever I am seeing in this milieu or whatever is my guess is true. If it is not, that it means it is much more complex than I am understanding. So, you see, we open up a problem, and this is how we have to understand that cell culture, tissue culture, explant culture, whatever you call it, these are acute systems. These are not, I mean, one has to be very cautious and careful that what kind of data are we interpreting out of it, because once they are in isolation, say for example, these cells, just let's talk about a reverse situation. These cells are not allowed, if this X, what are you talking about, this X thing, this X compound is secreted by this pink house to ensure that this does not form this kind of structure. It is not allowed to form this second roof. Now, I say, well, if this is true, then if I grow this in isolation, it should be able to form this roof, second roof. If this is true, then it means that X compound is important. And you can do the reverse experiment. You put X here, you should not be able to see this growth. So, you are realizing that what a profoundly powerful tool this could be, provided you ask the right question. And if you do this whole process, like just like it is another technique, I will just follow it without even putting my brain into place, you will end up with, of course, you will get some results. I am not denying that, but something which may be very ambiguous, and many other people may not be able to repeat it. So, one has to realize that before using a tool, one has to understand how close you are to the in vivo condition, or you do not care. There are people who use cells. So, there is another group of people, we will talk about them, who use cell as a simple bioreactor. Okay? What do? Say very simple. All of you have heard about antibodies, right? We get antibodies, production of antibodies. So, I say for example, these cell type, and I am representing the analogous, okay. These red cell types which are there, they produced antibodies, say, Y. What I do? I take out these things, I understand the conditions under which and I produce tons of antibodies for some z function. I can use the cell as a simple, in that situation I am using the cell as a simple bioreactor. And there are many such studies in biotechnology where people use them as bioreactor. They are just producing unit. As a matter of fact, a lot of oil production which is happening with the algae, we are using algae as a bioreactor because it is producing algae, we are isolating the oil and that is it. So, we are not really bothered about it. Our goal is very clear. We are using this as a tool, simple tool to produce some XYZ compound of our own commercial medical significance, full stop. Okay. So, unless you know what you are asking for, unless you are sure the question you are asking, and another thing here, when I talk about, when I was telling you this when I made this complex picture in front of you, I told you this is the real situation. So, how far is your culture? So, one thing you have to realize, fundamental question what you have to ask, how far or how close is the in vitro cell culture system with respect to the in vivo condition. This is one fundamental question one have to ask, provided if you are working on some biomedical problem. Okay. This is one situation. The next is two, using animal cells as sensors. What do I mean by sensors? Say for example, you can use, most of our cells are very sensitive to different kind of toxins. So, I have this culture dish, I put the toxin, I know how it reacts. So, based on that I am using this as a simple sensor device. So, for testing, for screening, or as some condition where I am not sure what this toxin is going to do, I use this as a simple sensor. So, I use the cell, if this is your cell, I use this as a sensor system. So, I have this compound, say X, Y, Z, whatever, and I have an output here to measure. What is the output? That could be electrical output, it could be chemical output, or it could be some other physical parameters within it. Okay. Similarly, I can use animal cells for producing or mass producing proteins or other biomolecules. So, in other words, I am using cells, giving them the right set of conditions, which first one has to study through cell culture, to produce biomolecules of my choice. So, you see, technique is one, but one has to ask the right set of questions. Or I study, there is another side which is previous to this one. If I put this as A, there is a B side. I use this as a regenerative medicine where which is one of the very fashionable topic currently in the stem cell differentiation and therapy. And which eventually may lead to artificial organs. Okay. So, one technique which most of the people consider technique, for me it is a philosophizing a subject. Unless you philosophize it properly, you will never be able to know the power of this, and it has tremendous power. Trust my word, it has tremendous power, but one has to know what you are doing. So the whole sole purpose of introducing a course which otherwise always believed as a simple, you go to a lab, you learn this from a senior, this is how you eat culture cell, that's it, that's just a technique. Let's come out of that mindset. Absolutely. Let's embrace this from a different world. Try to imagine we can create structures, we can create next generation sensors, we can create the cheapest sensors, we can produce our own setup antibodies in an artificial synthetic systems. Let's think beyond this, because let's use cells as a tool. We govern them, we tell them what we want it to do, we tell them then which part of the DNA we want to put, transcribed. Let's go take it to that level where we can truly say we are doing a cell engineering, and here is the platform where we can do the cell engineering right within these limitations. So, with this hope, we will be proceeding further in the next class. We will talk a little bit more about these conditions. We will take over from one of these pictures, that what all the forces, what you have to take into account. We haven't still talked about synthetic systems, in vivo systems, when you are translating, what all you needed. There is a biology behind this whole thing. What are those biological parameters? And I will try throughout the course, I will try to make it very generic, so that anybody who is the viewer of this course, whatsoever field they are, it should be tangible to them. So, bear with my using the simple languages instead of using technical jargons, which I am not going to use throughout the course. And if I use, I am going to write it down here. Okay? Thank you, and thanks for your patience listening.